Assalamualaikum, have a very good day. Today our group will present about SDA's page. So this is our group members: NURAMILA Sofia binti Jamal, Muhammad Akmal bin Muhammad SALIHIN, Muhammad FARIEZ bin JAMALLUDIN, NURNAJIHA binti NASRI, and SARA binti MOHAMED NAZRY. Introduction of the SDS page. SDS page, also known as sodium diadoxyl sulfate polyacrylamide gel electrophoresis, is a common method in biochemistry and molecular biology for separating proteins based on their molecular weight. It is frequently used for protein analysis, which includes protein identification, molecular weight determination, and purity evaluation. It is a useful technique for comprehending the composition and behavior of proteins since it can separate proteins under denaturing condition. Next, we will move to the principle of SDS page. According to the SDS page principle, when a charged molecule is exposed to an electric field, it migrates to electrode with the opposite sign. The relative mobility of charged specimens determine how the charged molecule separates. Because there is less resistance during electrophoresis, the smaller molecules migrate more quickly. The charge and structure of the proteins affect the migration rate as well. The proteins are separated according to the length of the polypeptide chain after the influence of the proteins. Charge and structure is eliminated by sodium diadoxyl sulfate and polyacrylamide. This is the list of instruments that used in SDS page. So for the apparatus, it used an electrophoresis chamber and power supply, glass plates, casting frame, casting stands, and combs. And for the materials, it used acrylamide solutions, isopropanol or distilled water, gel loading buffer, running buffer, staining solution, protein samples, and molecular weight markers. Next, this is a type of samples of SDS page.
So first, we're assembling the plate. The apparatus consists of glass plate, the casting frame, and also the casting stand. So by doing this, we can attach them first by combining the slides and then combine with the frame, and lastly combine with the casting stand. But make sure to ensure that the plate was free of leaks by adding water and checking if there are any leaks before we starting the experiment. And then we are ready to prepare the resolve gel preparation. First, we add double distilled water to ensure priority for optimal result. So where water acts as a solvent for our gel. And next, we add trace HCl buffer with a pH 8.8 to help maintaining a stable pH environment. And now, we mix with acrylamide mix, which is the blend of acrylamide and bisacrylamide. The ratio between this monomer determine the pore size of our gels. And next, we pour in sodium dodecyl sulfate or SDS, the detergent that is transforming these proteins, and unfold them and coating them with negative charge. And the next, we add in our catalyst where we combine with ammonium persulfate initiates the polymerization process. And last but not least, we're adding ammonium persulfate and our initiator, working hand with teammates, and it generates a free radical kicking starting the polymerization reaction. And next, after we prepare our resolving gel, we fill and we add to our casting frame until the level that we have been marked before. And don't worry if there contain a bubble, because after fill until the level remarked, we're gonna eliminate the bubble by using isopropanol, and we will wait for the gel until it's solidified. And next, for our stacking gel preparation, the material will be the same. just they will be different in terms of volume and also the different in pH for 3HCls because the stacking gel it need a higher pH to help create more uniform electric field and allowing proteins with different isoelectric point to focus on the narrow zone. So after we prepare our stacking gel, now we're gonna fill in our casting frame until it fill fully on the top of the casting frame. And before we let the gel stacking, we're gonna insert our comb and let it solidify. And after that, we're gonna continue, and when the gel is ready, to proceed to the next process.
Okay, what is electrophoresis? Electrophoresis is a laboratory technique used to separate DNA, RNA, or protein molecules based on size and electrical charge. SDS page gel is made out of polyacrylamide. Polyacrylamide gels are formed by reaction of acrylamide and bisacrylamide that is resulting in highly cross-linked gel. These are the components of SDS page. Samples are prepared by mixing it with denaturing loading dye in a small 1.5 ml centrifuge tube. Before loading into gel, the samples are placed in a heating block set at 95 degrees Celsius at 600 rpm for 5 minutes. While the samples are heating, the gel running apparatus is assembled. Samples are prepared by mixing it with denaturing loading dye. In this video, they are using precast gel. Remove the seal from the bottom of the precast gel and the cone on the top of the precast gel before use. In this video, they are using precast gel. Remove the seal from the bottom of the precast gel and the cone on the top of the precast gel before use. In this video, they are using precast gel. Remove the seal from the bottom of the precast gel and the cone on the top of the precast gel before use. In this video, they are using precast gel. Remove the seal from the bottom of the inner electrode assembly unit is removed to place the precast gel and the buffer dam plate to complete the assembly of it. The arm is folded down to place the gel and the buffer dam side by side. Press the gel and the dam towards the center and fold the arm back. Place it back in the tank. Make sure the tank is at the rear with the black electrode on the left. Inner electrode assembly unit is removed to place the precast gel and the buffer dam plate to complete the assembly of it. The arm is folded down to place the gel and the buffer dam side. The inner assembly forms inner chamber inside of the tank. Fill the tank with running buffer to the top and fill the outer chamber approximately to the two gel lines. Usually, 20 microliter of sample is loaded using micrometer pipette. The sample is slowly dispensed into the well between the two gel plates. Samples that is badly prepared, which is containing air bubbles, will cause insufficient samples being loaded, while the samples that is loaded very quickly will spill out from the well and will also result in an insufficient samples being loaded. The inner assembly forms inner chamber inside of the tank. Fill the tank with running buffer to the top and fill the outer chamber approximately to the two gel lines. Usually, 20 microliter of sample is loaded using micrometer pipette. The sample is slowly dispensed into the well between the two gel plates. Samples that is badly prepared, which is containing air bubbles, will cause insufficient. Once the sample is loaded, complete the assembly of the tank by placing the lid over the two electrodes, black to black, red to red, and press down. Connect the electrophoresis unit to the power supply, making sure black connects to black and red connects to red, and then switch on. Once the sample is loaded, complete the assembly.
Okay, firstly, in sample loading for SDS page, we will retrieve the sample extract from the freezer and thaw completely, and then we will vortex for about 10 seconds. Okay, firstly, in and then we will use micropipette tips to load up to 15 microliter for each sample into the wells. And then we load 5 microliter of molecular weight marker into one lane. Lastly, record the loading order for reference. And then
Hi, my name is NURNAJIHA. I will explain further about the conclusion. So, SDS page is a powerful tool for protein analysis. From gel casting and sample loading to electrophoresis and visualization, each step plays a critical role in achieving accurate protein separation. This method is essential in biochemistry, molecular biology, and clinical diagnostics for analyzing protein size, purity, and composition. So that's all for our video demonstration. Thank you for watching this presentation. We sincerely hope it has deepened your understanding of the SDS page process and its importance in scientific study. Thank you. Bye bye.